Say hi once you see me and hear my voice. Say hi once you can see me. Say hi. Say hi once you can see me and hear me. Say hi. So without wasting our time, we are going to the lecture today, which is going to be on the introduction to microbial genetics. This is going to be a beginner class with the assumption that you are just starting microbiology. So it's going to be a very brief one, and so that at the end of the day, you could as well go again and go and revise it. This lecture is titled Introduction to Microbial Genetics. Okay, the as a mean of introduction into this course, you are going to learn what microbial genetics is, why is microbial genetics, why is it so important, and how can it connect with other field of microbiology. For example, you should be able to understand some basic terms and concepts in microbial genetics. Then later you should be able to explain how microbial genetics, how microorganisms inherit and express their characters. Then you should be able to also relate microbial genetics to health, industry, and the environment. Microbial genetics is not just a genetics of the microbe, but it is the study of how microorganisms store, transmit, and express their genetic information. If there is an information, for example, special information like ability to adapt to a certain environment, ability to metabolize glucose, ability to do one to perform one function or another. For example, causing diseases, uh, and so on and so forth. So the how this microorganism, how would they store all these characters? Then how can they pass it from one microorganism to another, or how would they pass it from being a parasite to their host? So that is a transmission. And how will all this be expressed in terms of genetic information? So don't forget that microorganisms include bacteria, archaea, fungi, protozoa, and viruses. Then the genetic information in most microorganisms will either be primarily DNA, except in viruses where we have it in RNA form, so like retroviruses. So then microbial genetics also help us to understand the microbial behavior. How do they behave in certain environments? Then how will they survive and how do they adapt? So, and this is widely applicable in biotechnology, medicine, as well as in agriculture. What are these? We are going to introduce ourselves to basic terms and terminologies in microbial genetics. Don't forget that a gene is a segment of DNA which is coding for a particular RNA or a particular protein. So that segment of DNA is what is called a gene. And the second one is genome. This is a complete set of genetic material of an organism. Could it be microorganism or even any organism? The genome is the totality of the genetic material in that organism. Then DNA, which is the double, which is deoxyribonucleic acid. This is the primary form of genetic carrying information. So DNA is a molecule carrying genetic information. Then RNA is an intermediate between um, molecules of DNA as well as a protein. That's why RNA is ribonucleic acid. So then mutation, we are going to also, in this course, we will also talk about mutation, which is a sudden change in the DNA sequence, or what we can call genetic sequence, sudden change. Then plasmid, we will also come across what is called plasmid. Plasmid is nothing but a small extrachromosomal material that can be found in DNA. And research has told us that majority of microorganisms that has DNA, that has plasmid, they are more virulent. They cause more infection. They cause more havoc in terms of infection, and most of them are drug resistant. So wherever you see any microorganism, any bacteria containing plasmid, you will know that this is going to confer a special form of resistance or special form of character in that bacteria. Then in this course, I want to believe that you are still going to come across most of the things you have learned in microbial ecology. So don't forget that microbial ecology is the study of how microorganisms interact with environment and how do they interact with each other. So that's why most of what we have learned in STM 317 is still going to be very useful in this course of studying microbial genetics. Then microorganisms, they exist in communities. They don't exist alone. They exist in communities. You find them in soil, they are found in water as well as in the air. So, and some of them still live on another organism. So, and their way of interaction could be in form of beneficial, which is a mutualistic form of interaction. It could be neutral, that is when neither both party is gaining or losing. So that is neutral. That is, it is not going to cause any harm to either the host or the microorganism. Or in a case where microorganism cause the parasites, where microorganism cause more havoc is the association is called parasitism. So and microorganism recycle nutrients and maintain temperature. Okay, part of the importance of microbial ecology will include that it is very important in nutrient cycling, such as nitrogen fixation, carbon cycle, soil fertility, which thereby enrich the soil. Then some of them also assist in the human health by assisting in microbial biota, thereby helping human being to digest some certain vitamins, and as well these are very important also in immunity. And we will also need this in industry for fermentation processes, food processing, which is example of this is yogurt, that the production depend on microbial ecology, that is microbial interaction. Then you will remember that also in first semester did you offer a course which is called microbial physiology and metabolism. What do we need in microbial physiology that is still going to be applicable in this course? For example, don't forget that microbial physiology deal with how microbial cell functions. For example, study of cell structure and organization, then understanding how microorganisms obtain their energy, could it be autotrophic, and so on and so forth. So how do they grow and reproduce? Example is how E. coli ferment glucose. And I want you to also generate a visual, that is a bacterium cell. You should be able to draw a typical bacterial cell diagram so that you be able to remember. Then the cell structure in microorganism, don't forget, microorganisms are made up of key components which include their cell wall. This provides shapes and protection. Plasma membrane, which control the movement of substances in and out of microbial cell. Then the cytoplasm, this has contained numerous enzymes as well as molecules. Then nucleus, which is the location of the bacterial chromosome. Don't forget that microorganisms, especially bacteria, they are prokaryotic organisms, and that is telling you that there is not going to be anything like organelle. So you nothing like nucleus. So what you have is nucleoid, which is the location of a bacterial chromosome. Then just like I told you, the cell structure of microorganisms, some of them will contain plasmids, which I said is an extra chromosomal material that is found in the bacteria. Then ribosomes. When you come across ribosome is the site of protein synthesis, and you should be able to label prokaryotic cell diagram. Then metabolism include all biochemical reactions that will take place in a bacteria or in any microorganism. This includes catabolism, which is the breaking down of molecules to release energy, as well as anabolism, which is a synthesis of a complex molecule, that is the joining together of small molecules to make complex molecule. Catabolism is the breaking down of bigger molecule in order to get smaller ones. So metabolism will keep microorganisms alive as well as also helping them to be able to adapt. So later on, I want you to also pay attention to other videos in Omega Bio Nexus so that you'll be able to know what catabolism and anabolism are. ATP, this is the energy currency of the cell. That is, it's just like when you have dollar, when you have naira. ATP is the naira of the cell. So, which is adenosine triphosphate, is the energy currency of the cell. So, which is very, very important in glycolysis. That is the breaking down of glucose into pyruvic acid, and Krebs cycle, which is the complete metabolism of glucose into CO2 and water, and in form of oxidative phosphorylation, just like what we are going to see in plants. Then ATP also help in power biosynthesis. They also need ATP for motility, that is movement and transportation. So without ATP, microorganism cannot survive. So it's just like somebody that doesn't have any if you don't have naira, if you cannot spend naira, that means you will not be able to survive in this so it's able to generate a fissure, that is where you have ATP molecules diagram with arrow showing how energy is being released, and how energy is also being produced as well. So then also you also need to know that no metabolism can complete, or no metabolism can progress without enzymes. That's why enzymes are said to be protein that will act as a biological catalyst. So another thing is that enzyme will help to speed up biochemical reaction, and enzymes are highly specific to their substrate. They are very, very specific. It's just like when you have a key, when you have a lock, there are certain, not all keys that can open a door. So the same thing is applicable to enzymes as well. They are highly specific to certain substrates. Then example include amylase. Amylase break down starch. Protease, that means from protein it break down protein. Then the importance of enzyme, I told you that without enzyme no metabolism will continue, and it will not even progress forever. So you just discover that Somebody will just eat and nothing, if nothing, no enzyme is going to act on it, the person will just not digest anything at all. So that is the function of enzyme. Genetic material, which is in form of which is DNA. Genetic material determine traits in microorganism. So once you can get hold of the DNA or the genetic material in microorganism, you can readily predict or correctly predict what is going to be the traits of that microorganism. Is it going to be virulent? Is it going to be non-virulent? Can it adapt? Is it going to be autotrophic? The mode of living, the mode of feeding, and so on. Then bacteria usually have one circular chromosome. So the chromosome you have in bacteria is unlike what you have in eukaryotic organism. Their whole chromosome is circular, not in form of eggs. So plasmid carry extra genes. Wherever you see plasmid, I told you they are extra chromosomal materials. That is, they carry extra gene. There are certain genes or certain traits that cannot be found in the chromosomal DNA, but they can be found in the plasmid. For example, the characteristics of drug resistance are mainly inherent in plasmid, not even in the main DNA. So whenever you see bacteria possessing DNA, then having plasmid by its side, that's telling you that such microorganism is going to be resistant. There are certain genes that will be inherent in that plasmid for resistance. The structure of DNA, this is not the first time we'll be talking about the structure of DNA. DNA is made up of is a nucleotide which is arranged in double helix, and the bases in DNA are adenine, thymine, guanine, cytosine, and then the pair that is A will always pair with T, then C will always pair with G. This is what is called the charger rule. So strands are complementary in the sense that since it's a double-stranded molecule, once you have one strand, you should be able to predict what's going to be the content of the other strand. So for example, when you have ACT now, or when you have CAT, so you should know that the second strand is going to be GTC. So that is that. And this double helix was discovered by Watson and Crick in 1953. So you can show the DNA double helix, you can look for the diagram. Then RNA and its types. RNA is unlike DNA is single-stranded, and there are basically three types of messenger RNA. For example, is a messenger RNA, just like the name implies, messenger, because it carry code from DNA. That is why it is called messenger RNA. And ribosomal RNA, because it is part of the ribosomes, it helps in protein synthesis. And the last one, which is transfer RNA, carries amino acid to the ribosome. So together, they translate genetic information in order to uh, produce protein. So protein perform most cellular functions. Protein are the most abundant, they are the most needed because they form enzymes in form of transporters. They are also in form of being used for structural adaptation. Then it also allow microorganisms to be able to adapt to stress, to be able to survive in the presence of certain antibiotics. Protein are made up of amino acid. That is to tell you what protein can do in microorganism. So summarily, to tell you that microorganism cannot do without protein, because they cannot do without enzyme, they cannot do they cannot cope if there is no protein in them, especially in the presence of antibiotics and so on. Okay, and this is to remind you of of the central dogma theory in molecular biology. If you don't even know any other thing, central dogma theory is very, very important. So, and that is the translation or the arrow flow from DNA to RNA, then RNA to protein, and DNA is transcribed to protein. DNA is transcribed to RNA. Don't forget that the RNA is single-stranded, then the DNA is double-stranded, then to protein. So protein carry out the cellular function, just like I said. Then just like I said in the introductory part of this lecture, I said mutation is very, very important in this course. You have to understand mutation as changes in DNA sequence. So this can be neutral, it can be harmful, or it can be beneficial. Don't forget that in your HND1, I made emphasis on this, on point mutation, insertion, on types of mutation include point mutation. It can be in form of insertion, deletion, and so on. Then mutation, one of the reasons why mutation can be beneficial is that it will allow variation, it will allow for variability, and that is the first tool to evolution. Then causes of mutation may arise from spontaneous error. That is, spontaneous means that the normal error during DNA replication. Whenever the DNA is getting replicated, there are certain errors that is going to be copied along the line, and these are called spontaneous mutation. Then exposure to certain chemicals, for example, the exposure to certain mutagens. There are some substances that are mutagenic in nature. There are, for example, alcohol, ethanol, benzene. All these are called mutagens. And in some cases, like smoking, that is nicotine, could also cause mutation. These are examples of chemicals that can induce mutation in bacteria. This has been experimentally proven. And another cause of mutation include radiation. And this radiation could be in form of ultraviolet rays, X-rays, and some of them. So just like I said, that some mutations are advantageous. For example, the one that we confer antibiotic resistance. So there are some mutations that is going to help the microorganism to be able to resist certain drugs. So that means that kind of mutation is very, very advantageous for that microorganism. Then microbial adaptation. How will microorganism adapt to the environment? Bacteria will adapt genetically to changing environment through biofilm formation. You will see some of them, they will just continue to exist in community to form slippery substances, that is a biofilm. Then another one is it will be in form of spore formation. You will see some of them that they will have something to protect them, as if they are like a coconut, as if they are found in the shell. So spore formation, example is bacillus. Then gene regulation. I told us this is not the first time I told you that whenever you have microorganism that is carrying out physiological function or metabolic function, and that gene cannot be turned off. That means most of the energy will just be wasted. So gene regulation, we are going to see this by the time we also continue in this course. Don't forget that this is just the introductory lecture to microbial genetics. Okay. Then again in this course, we are going to make mention of horizontal gene transfer. Horizontal, that is one between one microorganism and another one, but they must be of the same species. That is horizontal, and it could be in form of conjugation, which is going to be through their pilus. It could be in form of transportation, that is going to be up uptake of DNA from the environment when microorganism must have died, and but the DNA will still remain, that it can also transform another microorganism. So transformation, then transduction, that is virus mediated transfer. We are going to see all this. The another one is a translocation, or what you can call duplication. We are going to see all this as we go on. Then I said plasmid are small circular DNA in bacteria. What do they do? They carry genes for antibiotic resistance or toxin. So it can be transferred from between bacteria. Then this is a powerful tool in biotechnology that we call cloning of vectors. So antibiotic resistance. We are also going to talk about antibiotic resistance, and because this one is a global health issue, is a threat to the world at large. So antibiotic resistance. Bacteria acquire resistant genes from plasmid. I said so. Resistant strain will spread from one hospital to another. So example is methicillin resistant Staphylococcus aureus. So we get to know this as well. Then microbial ecology and human health. Microorganisms live in, and some of them also live on the human body. Those that live on the inside human body, they are found in the gut because they aid digestion, as well as also helping us to be able to fight against a certain infection. That is telling us that they are also very important in immunity. Then they also, those ones that live or on the human being can also be in form of pathogen. That is, they can be pathogenic because they cause certain diseases. And some of us, many of us will know, can give examples of different types of microorganism and the different types of diseases that they can form. Then balance between helpful and harmful bacteria or microorganisms is very, very crucial. Both those ones that help in digestion and immunity, and those ones that also cause infections. So there should be a kind of balancing, there should be a kind of equilibrium at which both of them are going to be to coexist together without causing any problem. Then the industrial application of microbial genetics will include in antibiotic production. For example, is penicillin. So we will see that microbial genetics has actually offered itself to be used in production of certain antibiotics. Then also in enzyme production as well. Then also fermentation that we make use in yogurt, beer, and bread. Okay, so environmental application. Microorganisms help the environment in form of bioremediation, that is cleaning of pollutants and waste water treatment. Some of them are used as decomposer that they use in recycling the nutrients. The practical method in microbial genetics, for example, PCR, which amplify the DNA. I'm going to see more of this as we go on, and DNA sequencing, which determine how the base or the bases, nucleotide bases, are arranged. Then the other one is a CRISPR. So the same way you can edit manuscripts. When you are typing and you just make a mistake, you want to type agency, and you now put N after G. So the same way you can also edit some DNA in bacteria. So we call that CRISPR. There are certain enzymes that are actually for that. So we are going to, why should microbiology students study genetics? So these are part of the puzzle that you should answer by yourself. Then how might microbial genetics affect your career? So you are supposed to be here. Then the case study is when you have lactose operon in E. coli. We are going to see more of this as we go on. This is going to be under gene regulation. Then we are going to have simple experiment idea. So grow microorganism on agar plates. So then how can you those agar plates you can just put them like just two plates. You put one in the dark, then you put another one in the light. Then you will be able to record the growth differences between these two. So this part of hands-on activity on microbial genetics. So those the one you put in the light and the one you put in the dark, both of them will have to adapt to the environment. For example, if the normal way of existing should be in the light, that means the one you put in the dark should be able to develop certain or acquire certain characters in their DNA for them to be able to survive in the dark. So that's going to record the differences in terms of the color, in terms of morphological features, and so on and so forth. Then, what are the challenges that we face in studying microbial genetics? Number one is that mutation rate is unpredictable. You can never tell which area or which way that protein can proceed to. So then the spread of antibiotic resistance is a major threat to uh, to human health, just like I said. Then there are ethical debates in genetic engineering. Up to today, there are some people that still believe that manipulation of any enzymes or manipulation of microorganism DNA should not even be practiced at all. Oh, and that is what we call genetic engineering. Genetic engineering is nothing but manipulation of DNA to get your desired result. So we are going to see this as we also go on. Then in the future, there will be more attention will be shifted towards CRISPR, which is a gene editing that will allow us to be able to edit the gene of bacteria in the same way we want it to behave. Then synthetic biology is very going to be very useful. Then designer probiotic for health. There are certain microorganisms that have used in fermentation that we once we are able to manipulate them genetically, we can produce more of it so that we consume them more than those ones. So, revision, a quick recap, ask yourself, what is a gene? What do you mean by a gene? What do you mean by genome? What do you understand by DNA, RNA, mutation, plasmid, central dogma theory? These are part of this. Then you are expected to, okay, quiz, define genome, name two types of mutation. What is a plasmid? What do you understand by the central dogma theory? So, which concept is okay? Then conclusively, microbial genetics is a foundational to microbiology. That is, without understanding microbial genetics, without our proper understanding of how microorganisms store DNA or store their genetic material, how do they transmit it, and how would they also express it? So it's very important. That's one of the reasons why the first thing we are going to know is how are genes being expressed. Then we should be able to explain heredity in microorganism. Don't forget that heredity is how to pass some certain characters from parent to their offspring. How are they inherited? So, which is very crucial for medicine, very crucial in industry as well as the environment. Then, how will you be able to relate your knowledge in microbial genetics to your real world application? Then in the next type of lecture, it is expected that we will look at DNA and look at the replication of DNA in detail. Then we will also be able to recognize and list out the function of the enzyme that will be involved and the function of each of these. Then we'll be able to explain the process of transcription as as well as gene expression. So I have like a three quiz which I'm expecting you to attempt at the end of this lecture. Just attempt it and send your answer in the comment section. The first one is which molecule is the hereditary material in most microorganisms? Is it RNA, protein, DNA, or lipids? Then the essay type is explain microbial genetics. Why microbial genetics is important in medicine. That is the application of microbial genetics in medicine. Provide the answer in the comment section. Then the quiz two, which is going to be under microbial structure and function. Which of the following is not part of a bacterial cell? Is it ribosome? Is it cell wall? Is it nucleus? Or is it going to be cytoplasm? Then you have to describe the role of plasmid in bacteria, and you have to provide one example of their importance. Then genetic mechanism, which process involve the transfer of DNA bacteria using a pilus, transformation, conjugation, transduction, or mutation. So discuss how mutation can contribute to both microbial diversity as well as antibiotic resistance. So um, thank you for your listening attention. Don't forget that we are expected to react through the comment section and submit your comments in that area. So I don't know if anybody has any question. If you are still hearing me, please say hi in the comment section. If you are able to hear everything I said, please say hi. Say hi before you log out. Okay, I can see one person. Okay, please say hi. Okay, thank you. Thank you. I'm going to harvest the list of the attendance. The attendance will be generated automatically, and I'm going to harvest it, and I'm going to post it so that you'll be able to know your you'll be able to see your names and able to see about your attendance. Once again, I want to thank you for your attention. So the next time we will meet, I just look up for the notification. Okay.